Good afternoon. I would like to thank the organizers for the opportunity to speak today on spliceosome gene mutations. I have no disclosures. So 10 years ago, the landmark paper by Yoshida et al. Identified splicing factor mutations in myeloid neoplasms. And this was dominated by mutations in 4 genes, SF3B1, SRSF2, U2F1, and ZRSR2. And those are depicted below and enriched at the 3' splice site. Now, these 4 mutations that are commonly mutated are distributed widely across at least these 6 myeloid diseases represented here. And what we can see is, although no specific mutation is isolated to a specific disease type, there are enrichments, as shown with MDS, where SF3B1 is enriched, and CMML, where SRSF2 is enriched. But there's increased complexity that is not shown here. And that's because some of these mutations, some of these genes have mutation hotspots. And those include SF3B1 and U2F1, where there is more than one hotspot present in these genes. And so if we look deeper to see now if there's any disease association with these allele-specific or hotspot mutations, we see an additional layer of complexity. And we'll first look at SF3B1 and U2F1. And again, as I said, because SF3B1, as an example, has 2 dominant hotspot mutations at the K700 position and the K666 position, and that these are enriched in specific diseases. For instance, in MDS, the K700 variant dominates, whereas when we look at de novo AML and MPNs, this now switches to the K666 variant being the most dominant. And likewise, if we look at U2F1, there are 2 known hotspot variants that include positions S34 and the combined R156 and Q157 positions. Now, these are fairly equally distributed at the MDS disease state. But if we look at CMML and primary myelofibrosis, this is quite different, with, again, the R156, Q157 variant becoming dominant in those diseases. And in particular, in myelofibrosis, the U2F1, R156, Q157 hotspot is actually a high-risk mutation and has been included in the MIPS 70+ version 2 prognostic scoring system. So these 2 genes highlight a central theme of the talk today, which is that the hotspot mutations likely confer specific disease phenotypes. So instead of just having a U2F1 mutation, it is critical to know which type and what position the amino acid is mutated at. And so we also wanted to address whether these specific hotspot mutations confer hematopoiesis that is unique on its own. And so to do that, we looked at the existing mouse models. And there are 4 genes that are represented here, and each of them has mouse models that have been published over the past 10 years. And there's 2 genes that stand out to address this question. The first is SF3B1, because it does have more than one hotspot mutation. And the second is U2F1, which is uniquely positioned to address this question of, are there hematopoietic consequences of specific hotspot mutations? And as I said, these 2 variants are fairly equally represented at myelodysplastic syndrome, and an existing mouse model of S34F heterozygous knock-in had been published by Dennis Fay and Harold Varmus's group. So in collaboration with Dr. Fay and Varmus, we obtained the Q157 heterozygous knock-in mice and did a direct comparison to the existing S34F mutant mice to address these questions of similarities or differences in hematopoiesis, as well as the splicing targets. And what we identified was that the U2F1 S34F and the U2F1 Q157R induced distinct hematopoietic phenotypes in the blood. To study this, we studied the hematopoietic cell intrinsic effects of the mutations by taking donor bone marrow from one of the 3 genotypes, the control CREs, the S34F CRE mice that are indicated in purple, and the U2F1 Q157R heterozygous mice with MX CRE indicated in orange. We transplanted these donor bone marrow cells into lethally irradiated congenic recipients, and then after stable engraftment, activated the mutation with PIPC and monitored blood and marrow cellularity and distribution of counts. And what you can see in the peripheral blood is that the expected phenotype that Dennis had previously reported with a reduction in cellularity in the peripheral blood in the S34F mutant mice. Now, strikingly, the Q157R heterozygous mutant mice did not show this reduction in cell count and actually had a mild increase in the platelet count. If we look into the bone marrow, a similar distinct hematopoietic phenotypes are observed with a reduction in cell numbers for the S34F mice, but largely not for the Q157R mice. We went on to further functionally characterize the effect at the stem cell level by performing competitive bone marrow repopulation assays. And to do this, we again took donor marrow from each of the three genotypes and mixed it separately 1-to-1 with competitor wild type cells. These were then transplanted into lethally irradiated congenic recipients, and we monitored the mice for up to 24 weeks. And you can see, based on peripheral blood chimerism, that again, the U2F1 S34F mutant mice in purple that had received those cells had a competitive disadvantage relative to Q157R in the control mice. And so this differences in hematopoietic phenotypes was quite striking and unexpected between these two mutations that occur at similar frequencies in MDS. Mechanistically, we were interested in understanding if this was due to differences in splicing targets that were occurring in those cells. And to address this, we identified and isolated KLS cells from each of these genotypes and performed RNA sequencing. And what is shown at first is an unsupervised principal component analysis of alternative splicing of skipped exons. And we can see that there is segregation based on genotype here. If we then do a supervised analysis using RMATs to identify the junctions that are unique and shared between them, we find that the majority of these splicing targets are unique to each genotype with minimal overlap in shared junctions. And so based on this, we wanted to look back and say, well, are there critical splicing targets that confer the phenotype, in particular to the S34F mice, that is not present in the Q157R mice? And we focused on a specific splicing event in a variant H2A histone called H2AX. And importantly, we wanted to have the goal of trying to revert these phenotypes with rescue of these alternative splicing events in vivo. And so what we identified was these mutant allele-specific splicing alterations in H2FY that actually do impact hematopoiesis. So H2FY has two isoforms, H2FY 1.1 and 1.2, and these are from a mutually exclusive exon usage. And we can see that S34F reduces the expression of the 1.1 isoform. In the middle, you can see that the lower band representing the 1.1 in the gel electrophoresis is reduced in intensity compared to the wild type in Q157 mice. And this is then summarized in the bar graph below, where the S34F cells have reduction in the 1.1 isoform, but not in the Q157 mutants. Now, this gene, H2FY, is also developmentally regulated during B lymphopoiesis. This provided a way for us to functionally validate this in vivo, because remember, the S34F mice have reduced B cells in contrast to the Q157 P mice that do not have a reduction in the B cells. And so what we were able to do is re-express the 1.1 isoform in S34F cells, and we're able to rescue this reduction in B cell count. And so overexpression rescued the B cell count in vivo, importantly providing direct evidence of an altered splicing event that could rescue a phenotype in vivo. Now, the same isoform of H2FY 1.1 was also overexpressed in MDS cells and shown to increase erythroid differentiation. Again, nominating this as an important target that does alter hematopoiesis. Now, this critical event of re-expressing variants to try and rescue phenotypes in vivo was again shown recently with LZTR1 overexpression, which can revert a self-renewal phenotype in vivo in ZRSR2 knockout mice. And this is critical because it starts to get at stem cell function beyond just mature lineage cell counts, which we've shown with H2FY. And so moving forward, it will be critical to look at each of these individual allele-specific splicing alterations and understand what are the critical targets and how can we prove that they're critical by using in vivo systems for functional validation. And I think high throughput methods are going to be needed to attack this instead of going one isoform at a time. But these differences in isoform expression really highlight that this could be part of the underlying reason the phenotypes are different with some of these mutations. The other thing that could drive this is the co-mutation status, because remember, in patients, these are not occurring in isolation. And so to address this, we looked at the distribution of other mutations with U2F1 mutations, and we observed allele-specific co-mutation patterns. And what we see here is if we take a large cohort of MDS patients and look at the distribution of U2F1 S34F variants, U2F1 R156, Q157 variants, and overlap those with two genes that are enriched with U2F1 mutations in general, being AXXL1 and BCOR. But if we now look at how those segregate within the specific U2F1 mutant alleles, we see a very interesting pattern. And there's two groups of patients that you can see here. While BCOR overlaps significantly and co-occurs with U2F1, it is really almost uniquely enriched in the S34 group. And AXXL1, while it is enriched and co-occurs in U2F1 mutant patients, that enrichment is more skewed towards the R156, Q157 variant. And what we see now is these two groups of patients that do not share BCOR and AXXL1 in common may actually converge on a common downstream pathway called the PR-DUB complex, and that both sets of these patients could result in decreased monoubiquitinization on H2A K119. So this complexity of allele-specific mutations causing hematopoietic phenotypes, causing specific downstream RNA splicing that has been shown to have hematopoietic consequences, and that they have specific co-mutation patterns, really increases the complexity of this system. So when thinking of a splicing factor mutant patient, it will be critical to understand what mutation they have, not just the gene that they have mutated. So this may make it difficult to develop specific tailored treatments, but it turns out that there as many differences there as there are also some shared properties between these splicing factor mutant cells that actually create vulnerabilities that we could capitalize on. And so these shared vulnerabilities in spliceosome mutant cells, there's two that I'd like to highlight. So one is here, haploessential cancer genes. So the SF3B1, SRSF2, and U2F1 heterozygous mutant cells have all been shown to require a wild type allele to be viable. So these are haploessential genes. And by that I mean you have a cell with a heterozygous mutation and the wild type allele is also expressed and that cell is viable. But if you remove the wild type allele and have just mutant expression, that can lead to lethality. And that has been shown specifically in vivo models for SRSF2 and U2F1. And what we wanted to ask is, these were non-transformed cells. In a fully transformed leukemia cell, could the same property of lethality be present? And so what Brian did is developed a genetic model that allowed him to express the U2F1 S34F mutant either with the wild type or by removing the wild type. And he took and did secondary transplants of these primary AML cells into mice. And then he followed them. And you can see in the black curve, the control mice that received no doxycycline or PIPC, that they succumbed to their leukemia. In addition, if you just express the wild type using a transgenic doxycycline-inducible system, those animals also died of leukemia. And finally, the heterozygous S34F wild type expressing mice also had lethality. However, if we now take these mice and we turn down the wild type expression by removing doxycycline, you can see you've created a hemizygous state, and these AMLs are eradicated and the mice survive. And so this proof of principle idea of haploessentiality could be approached and tried to be targeted by modulating the ratio of these expressions, potentially with antisense oligonucleotides. The second shared vulnerability in spliceosome mutant cells is the induction of R loops. So all of these heterozygous mutant cells induce R loops, and R loops are a hybrid of RNA and DNA with a single strand of DNA. These occur in our normal cells, but are an increased presence in the presence of these mutations. And these R loops are normally cleared through an ATR-dependent process, thereby avoiding DNA damage if a replication fork is stalled. And so an interesting observation was that drugs that also induce R loops, which are splicing modulators and ATR inhibitors, have essentially a synthetic lethality with splicing factor mutant cells in preclinical models. So the idea that R loops are present in mutant cells, drugs further induce these R loops and lead to lethality. Really, it then raised the question for us, are there spliceosome mutant cells that are sensitive to other drugs that induce R loops? And so through a collaboration with Zongshen Yu's lab, who had the unexpected observation that inhibition of nonsense-mediated RNA decay, or NMD, can induce R loops, we went forward and addressed this question. And so what Zongshen Yu had identified was that in this very regulated process of mRNA decay, which requires SMG1 to phosphorylate UPF1 to orchestrate this, is that if you genetically manipulate SMG1 or UPF1, you actually can induce R loops. Julie Bayliss had an inhibitor to SMG1, and so Zongshen used this to show that the drug actually induces R loops as well. So S9.6 is an antibody that recognizes the R loops, and in the upper right, you can see by immunofluorescence that this treatment increases R loops, which is quantitated at the right. So using this drug, we asked, could SMG1 inhibition induce R loops and ultimately reduce viability in splicing factor mutant cells? And that is what was seen. So inhibition of NMD causes further increase in R loops and DNA damage in spliceosome mutant cells. So in U2F1, S34F expressing cells, you can see that the S9.6 intensity is increased, indicating R loop induction, shown here, and has been previously published. But now when we add the SMG1 inhibitor, you can see a further increase in R loop presence. Now, R loops are normally cleared and degraded through RNase H1. And so treating these cells with RNase H1 completely removes the presence of these R loops here, again, showing that what we're measuring is R loops. This induction in R loops, we also had a concomitant increase in gamma H2AX, or DNA damage in these cells that also can be abrogated with RNase H1 treatment. And the same observation was seen in SF3B1 K700E mutant cells. So this is not restricted to just U2F1 cells. Now, these induction of R loops and DNA damage also reduced the viability of cells. And remarkably, that this viability, the R loops are required for the hypersensitivity of spliceosome mutant cells to NMD inhibition. And this was shown using cell lines that expressed S34F or wild type U2F1. With increasing concentrations of SMG1, you can see that the S34F mutant cells have increased sensitivity compared in red compared to the wild type cells in blue. And by overexpressing RNase H1 in these cells in purple, you can again completely abrogate this reduced survival. This is also seen in SF3B1 K666N cells where RNase H1 reverts this reduced survival. So in summary, we've reviewed that SF3B1 and U2F1 have allele-specific hotspot mutations that are enriched in specific diseases. So moving beyond just the SF3B1 or U2F1 mutant status, knowing the allele-specific hotspot has importance. And that these U2F1, S34F, and Q157R mutations induce distinct hematopoietic phenotypes and splicing. That these U2F1 mutations also co-occur with distinct mutations that may converge on common pathways. And so the idea that this differences in alleles could drive specific phenotypes of splicing and be influenced by the co-mutations likely extends to additional mutations. And finally, the spliceosome mutant cells, even though they have diversity in the type of alleles that they're expressing, they may share some common vulnerabilities and may be sensitive to drugs that further induce R loops. And search for additional drugs that could do this could increase the approaches that we could take for patients with these mutations. So I'd like to acknowledge members from the Walter Lab that participated in these studies, the U Lab, as well as Tim Grobert, Andy Bruner at MGH, Dennis Fay and Harold Varmus at Cornell, and Julie Bayless at Amgen. And finally, this was supported from funds from the Edward P. Evans Foundation, the Taub Foundation, the Siteman Cancer Center, and NIH Spore and Leukemia. Thank you very much.